I don't know if I would promise a Benjamin Button effect using partial reprogramming anytime soon. On a cellular level, definitely. I mean, on a cellular level, reprogramming is the Benjamin Button effect, like to the extreme that you take an old skin cell and you reprogram it to an embryonic-like stem cell that's completely rejuvenated. And this is what happens, essentially, this is what happens after fertilization. Every time you have a new baby being developed after fertilization, this is what egg cell does after it starts dividing. There's complete rejuvenation of the 20, 30, 40-year-old egg cell that is the same age as the mother.
Yuri Deigin, welcome to Levity.
Thank you. Pleasure to be here.
I've been looking forward to talking to you. This is one of my favorite topics in aging biology, partial reprogramming. Yeah, I can imagine.
Yeah, absolutely.
We have a lot to talk about today, but maybe you could just give us a little bit of a background of who you are, where you come from, and how you got involved in this field.
Yeah, definitely. So I'm a drug developer, and I've been a drug developer for close to 15 years by now. And this is really how I got involved in the field. Through being a drug developer, eventually I got introduced to the concept of fighting aging, because the diseases I was working on are really consequences of aging. And this is something I wasn't aware of as I was doing my drug development stuff, and then made aware of by other fellow drug developers who were also by then also converted to fighting aging, and realizing aging is really the underlying cause of all of these diseases that we're trying to develop drugs against. And so really we're not being very efficient because we're just kind of playing whack-a-mole with the consequences of aging. Whereas if we really want to eradicate all these diseases and prevent all these diseases from happening, the most efficient way is to go after the root cause, the cave of the moles where they all live and breed, which is the aging process, because the aging process is what eventually causes all of these age-related diseases.
What year was this when you got when you realized that it's aging is the root cause and we should be focusing on that?
It was, I think, 2011, 2012 probably is more when I actually got introduced to this idea. This was actually Peter Feditchev played a major role. I'm sure many of people who are familiar with our space know Peter Feditchev quite well. And also probably the catalyst who introduced me to this concept of fighting aging was also Mikhail Batin, who actually is very well known and has been working on fighting aging as an activist, as an organizer for, I guess, close to 2 decades by now, if not more. And so it's kind of from that ecosystem that I got my first exposure to this concept of age research on aging and age-related diseases being the consequences of the aging process itself. And yeah, so this was around 2012, 2013. And then once I embraced this concept, I became kind of very passionate about it, and also tried to do what I could to advance this field forward. Initially as an activist, trying to popularize this idea, just to try to educate people to basically what I was ignorant of, that all these diseases that everybody agrees should be eradicated are consequences of aging, which surprisingly everybody doesn't agree should be done something about. So this cognitive dissonance of people, regular people, as I had, and it's very understandable. We're all just kind of raised with the idea that diseases are bad, but aging is natural. And so don't touch aging. You just be ready to age and die peacefully. But of course, cancer is bad and heart disease is bad. And, you're cancer survivor. We're all for fighting cancer. But when you say we're all fighting aging, you're like, wait, no, we're not. We're just diseases. Let's just eradicate diseases, but we're still going to die very healthy people, but at age like 100 is the limit. People have some sort of like very odd preconception that you just have to die because everybody else died, so you have to die. And I had the same thing. So it takes some time to kind of get over this uh, barriers in your mind that you just kind of, you're brought up to with this concept that you have to accept your mortality. And there's just actually trying to think about ways of fighting aging is somehow unnatural or bad or selfish or bad for humanity because it's going to cause overpopulation. So all of these misconceptions that I think need to be educated against, like the general public, before we all embrace, like society at large embraces that aging is a problem and we all should be striving to eradicate it and do so as quickly as possible. Until we can get over these preconceptions, I think we're still going to be a very small minority people who actually embrace this idea that aging is a bad thing and we need to be fighting it.
How did it? I mean, if we go back to 2012, how did it crystallize for you what you were actually going to spend your time on once you knew that this was where your focus was going to be?
It didn't. Yeah, for the longest time, nothing crystallized. Basically, it was two parallel things. I had my kind of work, drug development, kind of regular drug development, and I had my passion or kind of activism in the aging space, and they didn't really intersect. But the more I learned about the biology of aging and tried to go deeper, basically why aging happens, because first, you for kind of the first reaction when you hear about let's fight aging, you're like, okay, great, yeah, let's do it. And then you're like, so why does aging happen? And it turns out nobody knows. And like the actual state of knowledge of the space, the scientific space of age-related research is, at least to me, it seemed very incomplete. And so basically you have to do your own digging into the science, and that's what I have done. And for basically the conclusions I came to at that point were that there's a very strong component that epigenetics plays in aging. Basically, eventually I came to the conclusion that epigenetics is a key driver of aging. And sadly, there were no interventions that could actually work at the level of epigenetics. There were, at that time, like small molecules, rapamycin, of course, metformin was just the molecules that we knew extend lifespan in mice, and those were being popularized back in like 2013, 2014. But of course, their effects were very small, and I was kind of disappointed. I'm like, well, I don't want to get into this field for like a 15% life extension. I want, several times lifespan extension or orders of magnitude lifespan extension. And this is not what's going to get us there. Like small molecules or rapamycin at least or metformin are not going to get us there. So what else is there? How can we actually completely hack biology to stop the aging process in its tracks and maybe even reverse it? And at that point, I was trying to search for answers to that question, and I came to the conclusion that it's all about the gene expression patterns and their change with age, and potentially there's a program component to this change, whether it's purposeful program or an accidental program, like just a continuation of development. That still was an open question, remains a bit of an open question, but doesn't really matter because at the end of the day, I think if we are able to circumvent this epigenetic change or epigenetic program, we can get to this goal of significantly extending lifespan.
How did you arrive at that conclusion that epigenetics seemed to be so important in aging biology? I mean, today we have the work of David Sinclair, for example, and his popular book. But I mean, it hasn't been known for that long that epigenetics can be so important in aging biology.
Yeah, well, for me, it was much earlier than David's book has come out, and I had this concept, what I call the strong epigenetic theory of aging, and David's, I call it the weak epigenetic theory of aging, because And I was very happy, of course, to see David's book come out and him popularize this concept of epigenetics being a key driver of aging. But I think the difference we have with David is David thinks it's still this epigenetic change, or these epigenetic changes are still a consequence of some stochastic processes like DNA damage random DNA damage that cause these epigenetic machinery to get kind of removed from the places where they should be, to go fix the DNA damage, and this is what eventually results in epigenetic changes that drive aging. Whereas I thought and still think that the epigenetic changes that we see are actually programmed changes from the developmental processes that basically keep going with age and start causing bad things, basically aging. What begins as a developmental program then continues as an aging program. And this aging program, as is the developmental program, is carried out by epigenetics, by basically changes in gene expression that initially occur to grow the organism, basically change the gene expression of cells during growth, but eventually this program continues, and these changes, epigenetic changes, keep going and then start causing problems for our cells.
You're hinting very strongly now at what epigenetics really is. So epi is something on genetics, right? Should we just start from scratch and explain to people what epigenetics is?
Yeah, sure. Yeah, if people don't know this by now, yes, we should definitely explain what epigenetics.
Let's be inclusive.
Is as a concept. Yeah, definitely. So it's just control of gene expression. Yeah, and you're right. Epi means above, and epigenetics means above genetics, something that controls the genes. And why do we need to control our genes? Is because we are multicellular organisms, and we have very different cell types. And each cell in its nucleus has identical DNA. In all of our trillions of cells, but since we have different cell types, like 200, close to 200 different cell types, each particular cell type really needs a different set of genes to be turned on and off to actually make it whatever cell type it has to be, like a skin cell or a brain cell. So basically, a skin cell needs to express very different genes, or a few very different. Of course, there's some common genes between all cell types, but what makes those cell types unique is the different combinations of genes that are only turned on in that particular cell type and are not turned on in a different cell type. And so to control all these genes, you need mechanisms of epigenetics that basically takes care of this. It turns genes on, turns them off, upregulates them, downregulates them, basically controls their volume level, as sometimes I make that analogy. So epigenetics is a broad term that just means controlling different genes at different time points or different cell types, etc. And of course, we as an aging field, is interested in the changes with time, the epigenetic changes. Basically, why do same cells have a difference in their gene expression pattern? Like a young cell has a different pattern of gene expression than an old cell. And moreover, there are some very conserved changes that occur between all animals, all humans, all mice of the same age. Like a young mouse, young cells of a young mouse exhibit a very similar pattern of, regardless of which mouse it is, all young mice have a very similar pattern of gene expression, and all old mice have, again, a very similar pattern of gene expression, so much so that you can actually have clocks, a clock of transcriptomic clock or epigenetic clock or proteomic clock, a clock of basically what things change in a cell with aging in a very predictable manner, you could say a programmed manner. And it is actually these observations about these clocks that initially made me convinced, or made me strongly suspect, that there is this epigenetic component to aging, or gene expression component to aging, because it's just different steps. Like epigenetics is above the transcriptomic level, and then proteomic level is just different levels of what happens in the cell. Basically, everything then trickles down to the proteins. It's the proteins that do they actually work? And the levels of different proteins change, but what controls these levels is at the top in the cellular machinery is epigenetic changes, basically what genes get expressed and at what levels. And this changes with age. And these clocks that I mentioned that we have, these epigenetic clocks that were, I think, started to get popularized, like 2013 was Steve Horwath paper was published in 2014. This is the time that I think was forming my understanding of aging, because this is the time I was actively digging into like the aging biology. And probably that was kind of the fertile soil into which these seeds were planted and made me then believe that aging is driven by epigenetics. And
what is a clock? What
in
is
general,
an aging clock?
yeah. So I mean, from as a like an aging biomarker, basically, it's some predictable set of information based on which you can derive then the age of a cell, or at least a close enough estimate of age of the cell and the owner of that cell. So from a, like if we're talking about epigenetic clocks, it's the combination of a few of these sites in the genome, or a lot of these sites in the genome. If we're talking Horwath clock, it's 353 CPG sites in that original clock that basically are have the same pattern of changes between a young cell and an old cell, and even back in the development, like the cells undergoing development and embryogenesis, the same sites in the genome show the developmental process according to that clock. You can actually kind of track the age all the way back to the zero in an embryo, or within the aging process, you can accurately pinpoint, like if you just take a random cell from a random organism, person, or animal, and just check its epigenetic age, then you will see that it corresponds very closely to the actual chronological age of that organism. Or now you actually have clocks that are built to actually predict mortality. So if you take that cell from a healthy animal and check its biological age, you will see that it's a little bit lower than the average for this kind of chronological age. If you take it from an unhealthy animal or unhealthy person with some diseases, you will see that its biological age, or her biological age, is actually higher. So they're like biologically older than their chronological age, which means they have a higher risk of mortality than basically their people of the same age.
How are we to understand the idea of volume?
Volume in terms of gene expression, well, it's just different levels of the genes that get expressed, like how much of the eventual protein it gets made for a particular gene. And as an intermediate level, there's a level of the transcripts, the mRNA, basically how much mRNA for a particular gene gets produced in a cell. And with age, we see that a change in the level of proteins for a particular gene. Some genes get upregulated, usually like some bad genes like retrotransposons, for example, or transposable elements that wreak havoc on our genome when they get active in the cell. Their levels actually grow with aging, and some good genes, like some useful genes that do, for example, the machinery to repair misfolded proteins, for example, we see with aging their levels actually decrease. So that's what I call like the volume knob of the genes. Like the good genes get turned down, the bad genes get turned up. It's a bit of a crude metaphor, but
you hear sometimes the metaphor of the conductor who decides who's playing, so to speak, and how loud. What do you think about that image?
Yeah, I think it's also a good metaphor, and I think there's definitely a conductor, centralized control mechanisms for our daily functioning. The hypothalamus, I think, is our conductor of our daily function, from like circadian rhythms and body temperature and even hunger and all of these things. There's a central conductor, and hormonal, of course, regulation. Also within each cell, potentially there are some like responses to this centralized synchronization, including circadian rhythms. Of course, there's like circadian written drivers within the cell that get synchronized to the central driver, guided written driver.
Just a comment on what you said about the clock. So are you saying that there's a similarity between, for example, a mouse who's like 3/4 into its life and a human cell that's 3/4 into a human life? Is that how we should understand it?
Yeah, very much so. Basically, since the 2013 start on work on these clocks, Steve Horvath, he started back then. He developed, since after that, many different variations of these methylation clocks, these epigenetic clocks, one of which is this pan-mammalian clock, where he actually found some evolutionary conserved sites in the genomes between a mouse, a human, a whale, and many other mammals, where it's essentially the same site in the genes that is then used in his clock, in this pan-mammalian clock. And this pan-mammalian clock can be used to accurately predict age or get the age of the cells in a mouse and a human. And so this clock very accurately tracks the aging trajectory of like 300 different species of mammals, save for the speed. Basically, yeah, like if you normalize the life history of all these mammals between like a 0 and a 1, where 1 is like maximal lifespan and 0 is birth, then this trajectory is essentially the same. And of course, and also a very interesting observation is that among all of the mammals studied, or virtually all of the mammals studied, it's the first 10% of the life history of our like lifespan. The first 10% is spent on development, where this clock ticks an exponentially quicker pace versus the remaining 90%, where it ticks linearly. So like it seems that the first 10% is just accelerated, as we normally observe that all of our processes are accelerated during our childhood and development, and then everything slows down but still keeps ticking. And this is a very interesting observation, because to me it suggests that aging in mammals is very evolutionarily conserved process, except of course for the ticking speed, the speed at which it occurs. And the pan-mammalian clocks that Steve Horvath developed, for me, are an additional piece of evidence in support of that hypothesis, that there's a conserved aging / development program that begins as development but then translates into aging over time.
So we've laid a good foundation now with epigenetics, and we're slowly going to move over to partial reprogramming. And before we do that, I think we need to explain what full reprogramming is, and the Nobel Prize that laid the foundation for that.
Do it.
Yeah, I'm counting on you, Yuri.
Okay, I'll try my best. So full reprogramming is, as you rightfully point out, is what got the Nobel Prize in 2012 to Shinya Yamanaka, who discovered, together with his postdoc Takahashi, this process of reprogramming, where you could take virtually any cell and then reprogram it all the way back to this ground state of cellular development called embryonic stem cell, where everything in our body basically started from this embryonic stem cell when we were starting our life as a fertilized egg. That fertilized egg started dividing and gave rise to this population of embryonic stem cells that eventually form the entire organism. And there's some other tissues in some other cell types in this fertilized egg that take care of the placenta, but basically everything else that comes from the embryonic stem cell is what eventually becomes fully formed human or fully formed mammal or some other organism. And before the discovery of Yamanaka, basically the dogma was that this process of differentiation from an embryonic stem cell all the way back to some other cell types like skin cell or brain cell is an irreversible process, a one-directional process, where everything kind of rolls down this landscape, the Waddington landscape it was called, to terminally differentiated state, like a skin cell, but could never roll back up. And Yamanaka and Takahashi actually have disproven this dogma and show that actually this is a reversible process. You can actually take a skin cell and reprogram it all the way back to this ground state of development, this embryonic stem cell. And what then was noticed by our field, the longevity field or aging research field, was a very nice side effect of this process is that the cells that get reprogrammed are also rejuvenated, physiologically rejuvenated. And all the cellular hallmarks of aging that you observe in the starting cell are gone. Basically, the cell after reprogramming, if you then say reprogram it again to the same cell type that you started with before, so if you take a skin cell, reprogram it to an embryonic stem cell, and then again reprogram it to again a stem cell, skin cell. Sorry, this new skin cell is a young skin cell versus say a 60-year-old skin cell or 80-year-old skin cell, and all of the cellular hallmarks that you had in an old cell in this new cell are gone. So this was very nice.
This is not only an epigenetic change, right? You said all of the hallmarks are gone. There's a broad spectrum of effects when you do that, right?
Yeah, absolutely. So basically, not only, epigenetics is not even, it's a completely established fact that epigenetically the cell is rejuvenated to the level of an embryonic stem cell, because the epigenetic pattern and the pattern of gene expression in this pluripotent stem cell is that is observed in embryonic stem cells, which are, of course, the base level of epigenetics in our development. But also physiologically, like we have a bunch of hallmarks of aging, cellular hallmarks of aging, that we observe during aging that get accumulated in the cell, basically like double-stranded DNA breaks, number of DNA breaks, the nuclear envelope, misfolded proteins, mitochondrial function gets worse with age. But after reprogramming, all of those hallmarks are no longer there, or they're at the level of a very young cell. So you get an improved mitochondrial function, you get no misfolded protein aggregates in these reprogrammed cells after reprogramming. So basically the cell is rejuvenated, obviously epigenetically, but also more importantly, physiologically for us. If we're continuing kind of with the history of how we came to have partial reprogramming, is that once researchers in the aging field have observed this very fortuitous side effect of reprogramming, that it rejuvenates reprogrammed cells, then they started to wonder, okay, can we somehow harness this rejuvenating power of reprogramming and use it, as I said, like in vivo, to be able to rejuvenate already formed organisms, rejuvenate adult cells. And I guess, the first attempt to do that was the paper from the Serrano group in 2013, where they just created this transgenic mouse model in which you could activate Yamanaka factor genes on demand. And once the mice have reached adulthood, they activated those genes and just kind of wanted to see what happens. And the way they activated them, they just basically started to express Yamanaka genes continuously. And this first kind of most obvious way to do this, unfortunately, was not successful because the mice just died. And they started dying pretty quickly. Basically, by the second week of expression of these Yamanaka factors, they started to feel worse and eventually died. And they also started to develop these teratomas, these tumors, thankfully, not very invasive tumors, but still they don't look very nice. And when they reported this finding in their paper and had those pictures of teratomas in the mice, I think it had a very unfortunate effect that a lot of people right away started associating reprogramming with tumors, the teratomas, and by then I think it got a very bad rep that, Yamanaka factors in vivo, dangerous, cancer, bad, tumors. Okay, no, it's not promising. But thankfully, there was another group that was trying this in vivo, the group of Juan Carlos Espiso Belmonte out of Salk Institute in San Diego, with Alejandro Campo, who's leading this research effort as a postdoc in the Salk Institute. And they had a very similar idea that created a transgenic mouse model in which you could induce Yamanaka factors on demand with an external molecule like a doxycycline or tetracycline. And their first order of business was to try to see, okay, but what is the safe duration of this expression of Yamanaka factors that the mice can actually tolerate and not die? And so when they explored this, they found that it's actually quite short. By day 3, actually, mice start exhibiting some abnormalities, and actually some of them start dying. So really, if you want to be safe, 2 days is probably the safest, the longest you should be expressing these factors for in these mice. And that's what they've done. Then they decided, okay, let's just try expressing Yamanaka factors for 2 days, and then we're going to stop. And then we can do it on a weekly basis or some other repeated basis. They just then eventually just decided to do it on a weekly basis and see what happens. And what happened was that actually worked. That was both safe and it was therapeutically effective in that the mice lived longer. And of course, I should have mentioned that they used the progeric mouse model. The mice that had progeria with a genetic defect that made them live much shorter lives than normal mice. But it's quite commonly used in our field because it's just very efficient in terms of doing lifespan studies, because you have a very short turnaround time. Those mice live for a rather short period, and so you can have much quicker experiments with lifespan, trying to see does your intervention work or not. And so they used this approach on these transgenic mice, and they observed that using partial reprogramming in this mouse model made them live up to like 50% longer. And also not only that, but on the level of biomarkers or tissue histology, those mice looked better than the control group. Their tissues looked better. They had fewer senescent cells. They had better mitochondrial function, etc. And so this result then, once they published, forms now the basis of our partial reprogramming field, because it was a very encouraging result, and many groups initially didn't believe it. They thought it's too good to be true. But then once some groups reproduce some aspect of it, including lifespan increase, I think Jean-Marc Lemetre's team from INSERM then validated this finding of Ocampo of using partial reprogramming to extend lifespan in both progeric and actually wild-type mice. Then the field kind of got more and more optimistic, and by now started embracing this partial reprogramming approach. And by now, of course, everybody embraces it to some degree. I mean, there's always skeptics, but I think now they're in the minority of skeptics of partial reprogramming.
Yuri, I just wanted to, I mean, in some sense, it can be hard for people to really understand what this meant for aging biology and the people working in the longevity field, because I think David Sinclair, again, does a pretty good job of, maybe he's probably hyping it too much, but still he's talking about, the Benjamin Button scenario where you actually get rejuvenated, and he's talking, of course, about an entire person, and so we're not even close to be there yet. But this is the allure of this field, right? That you kind of can imagine that you could actually get biologically younger again. We don't have to go all the way there, of course, but still, this is kind of it's part of why it's so fascinating. No?
I mean, yeah, absolutely. And yeah, I mean, David Sinclair, he's definitely a PR savvy scientist who sometimes he kind of turns it up to 11, his kind of hype meter, that I would probably keep it at like a 5 or a 6 myself. And yeah, I don't know if I would promise a Benjamin Button effect using partial reprogramming anytime soon. On a cellular level, definitely. I mean, on a cellular level, reprogramming is the Benjamin Button effect, like to the extreme, that you take an old skin cell and you reprogram it to an embryonic-like stem cell that's completely rejuvenated. And this is what happens, essentially, this is what happens after fertilization. Every time you have a new baby being developed after fertilization, this is what egg cell does after it starts dividing. There's complete rejuvenation of the 20, 30, 40-year-old egg cell that is the same age as the mother. But on the level of translating these findings into therapies that people can use in the foreseeable future in the next couple of decades, I don't know if I would promise like a significant rejuvenation that an 80-year-old person will become a 20-year-old person, like Benjamin Button, like. But I think maybe we can expect with prolonged application of this therapy, like if a 50-year-old person starts using this therapy, and once we have a systemic therapy that targets like a bunch of organs, key organs that can provide, systemic rejuvenation, if that person at age 50 starts using on a repeated basis this therapy, I think we can confidently expect this therapy to allow this 50-year-old person to remain biologically much younger, that say by age 90, if he or she continuously uses this therapy, their biological age could remain, well, if maybe become 60. So maybe 90 will become the new 60, or even possibly remain 50 if this therapy is that efficient that it's completely freezes their aging process. Basically, it remains at the same level. It does not increase. Their biological age does not increase. And I think it will be a very beneficial, nice to have if we can actually reduce biological age. So maybe this 50-year-old person can actually reduce their biological age, say to age 40, once they start using this therapy. And as time goes on, their biological age can actually remain significantly reduced. I mean, I think it's hard to speculate with. I mean, it's easy to speculate, but I think it's hard to be at the same time very cautious with our optimism, because it all comes with a very wide confidence interval. I mean, it's possible that we might be lucky in our ability to translate partial reprogramming on various different cell types, that we will get to this therapy quickly that will allow us to have a significant reduction in biological age. But it's also possible that partial reprogramming would actually be tougher to translate than we anticipate, and it might not be as effective or as safe to be able to create therapies like that in the immediate future. Maybe 30, 40 years down the road, it's possible, but you have to be careful with what you promise. But I'm optimistic that we're actually somewhere in between this confidence interval and maybe closer to the optimistic side. But I wouldn't, as I say sometimes, I prefer to underpromise and overdeliver than the opposite. So
yeah. So correct me if I'm wrong here, but you were you were actually one of the first biotech startups in this field, right? The first
in 2017,
biotech startup.
In partial reprogramming, translating partial reprogramming, I founded the first company dedicated to this, in 2017. It was called Epigenetics. And the idea was to create therapies based on partial reprogramming because nobody else was doing it. And by in 2018, about a year after I started, then David Sinclair joined with YouthBio, and actually TurnBio, I think, was even earlier. Also in early 2018, they started Vittorio Sebastiano and Jay Sarkar out of Stanford. They started their company to translate their variation of partial reprogramming, where they use 6 factors, basically Yamanaka factors and 2 more factors. And David Sinclair joined the field with YouthBio, I think based on research of Yongcheng Ryan Liu, where he investigated using 3 of the 4 Yamanaka factors in this nerve crush model of vision loss. And so
what happened in that study? They crushed the optic nerve of mice, and then what happened?
Yeah, David has a very elegant story around that research and their findings. And basically they crushed the optic nerve, and then they used partial reprogramming to see if they can restore it and restore vision. And they succeeded, and they were able to restore vision versus control mice in which vision was not restored. But using partial reprogramming in that nerve crush model, they were able to restore vision. those mice. And this was, I guess, a watershed moment for David Sinclair. And then he founded LifeBio. Well, no, not LifeBio. The LifeBio was a conglomerate that meant to have a separate company for each hallmark of aging. And under LifeBio, there was Idune company that was dedicated to partial reprogramming. I think now it got rolled into LifeBio, which in LifeBio, I think now exclusively does partial reprogramming, or nearly exclusively does partial reprogramming. But yeah, initially it was based on, I guess, Yuan Cheng's or Ryan Liu's work, or I mean, jointly under David Sinclair's lab. There were a few other scientists working on it, including some collaboration with Vadim Gladyshev group. Bonhan Zhang, I think, also participated in some aspects of partial reprogramming with David's lab. But yeah, that's the story. And well, interestingly, like this OSK concept is also used by another company called Rejuvenate Bio. This is Noah Davidson. He also hails from Harvard. He was jointly in David Sinclair's lab and George Church's lab, or he worked with both of them. And recently they also reported some very cool findings of using partial reprogramming to extend lifespan in normal mice, in wild-type mice, which for the longest time has been kind of a main point of criticism for the skeptics of partial reprogramming. Have been saying that, oh, we still haven't seen any meaningful lifespan extension in wild-type mice. We've seen it in progeric mice, but maybe partial reprogramming only works in progeric mice, or at least in lifespan extension only works in progeric mice. But then Rejuvenate Bio has put out this very cool paper where they demonstrated that even in very old mice, they took like 124-week-old mice, which, if you compare it to humans, is like an 80-year-old human, and they dosed them with this gene therapy, this OSK gene therapy, same as David Sinclair's gene therapy, just the 3 Yamanaka factors, using an AAV system, with a split AAV system, same system that David Sinclair is going to take to the clinic next year for eye indication. But using the same system, this double AAV vector with OSK, inducible OSK, they extended lifespan, remaining lifespan by like 109%, but overall lifespan by like 7.5%, like close to 8%. Basically an extra 10 weeks, close to 10 weeks of lifespan. But which is still, I think, is a impressive finding, impressive result, because these were very old mice, as I said. Like it's basically, taking an 8-year-old person and doubling their remaining lifespan. And for a human, the remaining lifespan of a 8-year-old person is, I would say, probably on the order of 5 years. So like 85, I guess, because you have a 1 in 10 chance of dying. Well, actually, I guess, what am I saying? If it's a 1 in 10 chance of dying, expected lifespan is 10 years, like so 90. But of course, there's still an exponential increase. So probably actual lifespan, expected lifespan for an 8-year-old person, remaining lifespan is 5 years. But regardless, it's still an impressive result. But also now we have Altos Labs coming out, at least in a public interview by Rick Klausner, the chief scientist of Altos Labs, and saying they were able to extend lifespan in normal mice using partial reprogramming by up to like 25% increase in average lifespan, which is way more than 7.5 that Rejuvenate Bio has reported. So, of course, that still hasn't been published. We kind of keep our fingers crossed to see what the Altos Labs publication will actually reveal. But still, again, we are very, very optimistic in terms of like new results that keep coming out of the partial reprogramming field.
And Altos Labs is, of course, the very famous biotech startup that I think it's the most funded biotech startup in history, or something like that.
Yeah, they definitely have that crown. Well, I don't know, maybe now OpenAI with their billions, but yeah, they're definitely biotech startup. Yeah, $3 billion. Yeah, that's quite impressive.
So you mentioned AAV. Could you just spend a minute on explaining how these transcription factors are delivered and activated in cells?
Yeah, so There's several different modalities of delivery. The quickest, easiest one to start with is just mRNAs, which is what TurnBio does. TurnBio just delivers mRNAs for each transcription factors, and once mRNA is in the cell, it starts getting translated into a protein, and the proteins do the work. And I guess TurnBio tries to formulate their mRNAs that they only live for a short duration in the cell and get degraded within a few days. And this is what ensures the partial aspect of reprogramming. As you remember, reprogramming really needs to be kind of tightly controlled so as not to cause problems. Because, as I said, if you keep doing it longer than 3 days, at least in a transgenic mouse model where these factors are expressed in all cells, mice start dying after like 3 days of consecutive expression of Yamanaka factors. And so you have to formulate your delivery system to ensure that you can either control in a tight way this expression, or that it gets degraded also in a predictable fashion. So besides mRNA, which TurnBio is the only company, to my knowledge, that does the mRNA approach, everybody else does some sort of inducible system, which is what we use, which is what David Sinclair uses, where, like in the Ocampo study, you have a doxycycline inducible expression of the Yamanaka factor cassette. Without this outside molecule that you then give to the animal, these factors are not expressed. There's no reprogramming done until you give the mice or humans this molecule. In our case, it's doxycycline because it's a doxycycline cassette, but it could be any inducible gene expression system. There's several available. And so the way we package it, we use a lentiviral system because you need a lot of space for the cargo. You got to pack the 4 Yamanaka factors in your cassette, and you got to pack this tetracycline inducible cassette also in your therapeutic vector. And AAVs are too small for that. That's why David Sinclair and others and Ryan initially had to split their system. And also they use 3 factors. I don't think 4 factors will fit in a single AAV. So basically they have 3 factors, OSK, that they put in one AAV, and the tetracycline TET-on system that they put into another AAV, and they just deliver basically in the formulation, they mix the two and they deliver that. And only those cells, they got copies of at least one of each different AAV, then they can express the OSK in those cells. And so basically that's the idea. If the cell gets, in our case, our vector, which has both the TET-on system and the whatever we want to control, the OSKM genes, Yamanaka factor genes, then once we give the mouse, or eventually human, hopefully, tetracycline or doxycycline, then the genes start getting expressed. Yamanaka factors start getting expressed, and they start doing their work in epigenetically remodeling the cell, and epigenetically rejuvenating it, and basically bringing on this rejuvenation that partial reprogramming is meant to bring on. And then once we stop giving doxycycline, this expression ceases eventually, and this is what ensures this controlled duration of Yamanaka factor expression. So hopefully, I explained it.
Well, I was thinking, what are the therapeutic uses most studied these days?
Oh, I think by now there have been multiple papers in multiple disease models. Besides lifespan, of course, there was also the study by Rejuvenate Bio. There was another study that I mentioned by Jean-Marc Lemaitre, after, of course, the original Ocampo study that looked at lifespan. I think Altos is also looking at lifespan, but also much, I think, more broadly, or in terms of therapeutic benefit, there are more papers that looked for benefits of partial reprogramming in the context of some disease, including, for example, heart disease or eye disease. I mentioned David Sinclair initially looked at this very artificial nerve crush model, but then he looked at much more actual diseases like glaucoma and this NION indication, which is an eye stroke. Basically, you get vision loss because of blood vessel occlusion in the eye. And they've also observed therapeutic benefits that partial reprogramming can restore vision in the mouse model of glaucoma, in a mouse model of Nion, and actually now in a primate model, non-human primate model of this eye stroke, Nion. And this is the indication that they're claiming to start a clinical trial, or planning to start a clinical trial next year, David Sinclair's YouthBio. And we are looking at the brain at YouthBio. We, from the very beginning, we wanted, the brain was our primary target organ. And actually, the entire concept behind YouthBio is tissue-specific partial reprogramming. And how we arrived there is that very early on, actually together with Alejandro Ocampo, we arrived at this realization that partial reprogramming needs to be tissue-specific. Because first and foremost, there are some tissues, some organs that really need to be avoided from partial reprogramming because they actually cause the death of the animals. They cause the most problems during reprogramming when you keep expressing those factors. And this has been experimentally verified by Alejandro Ocampo in like 2023 publication, where they have shown that the liver and the small intestine are the two key organs that cause the most problems during reprogramming. And if you actually avoid expression of Yamanaka factors in those organs using a transgenic mouse model, you can greatly increase the safety window of partial reprogramming from like 4 days to 10 days of consecutive expression that all mice survive. So basically, if you don't avoid those two organs, then mice start dying, I mentioned day 3 or day 4. But if you avoid the liver and intestine, all mice survive for 10 days of consecutive expression of these factors.
Yuri, can I ask you there? You mentioned the liver and the intestines.
Sure.
So if you don't express, if you don't use partial reprogramming in those tissues, you could go for 10 days. So you get more of a rejuvenation effect that way. Is it like a gradually increasing rejuvenation effect the more the longer you go for, so to speak? You can get closer to the pluripotent end station if you avoid certain tissues.
I don't want to get to the pluripotent, closer
of
to
course
pluripotency,
not.
yes. And whether you get a better rejuvenating effect or not, it's still an open research question. Theoretically, I think you might. There's some indications to that end. And this is actually a area of ongoing research, including by Alejandro Ocampo, who now, he mentioned his next goal is to actually kind of beat the lifespan increase in normal mice. And one of the ways that he is planning to do so is by creating a transgenic mouse model that avoids the problematic tissues, definitely liver, small intestine, and some others, and maybe some other clever ways of minimizing risks of pushing the cells too far with reprogramming. But yeah, once you exclude those tissues, you can actually keep expressing those Yamanaka factors for longer with potentially better or stronger rejuvenating effects from those factors. And that's definitely one of the ideas of why we want to avoid those tissues and why we want to be tissue specific, because also this means that once we figure out what's the optimal duration of reprogramming for a particular cell type or a particular organ, we can then have very different protocols for different organs. If we, of course, we need to use different inducer molecules, and this is a planned area of research for us, where we can target. So for the brain, we crank it up to 11. We do a lot, because we can push neurons, and this is another reason for why we're focusing on neurons. We can push neurons pretty far with expressing Yamanaka factors, and they remain neurons. They keep doing their job, which is actually like the key problem with reprogramming is not that you get tumors or teratomas. The key problem is that if you push the cell too far, it actually stops doing its work or stops doing it efficiently. And if you push the liver too far and it stops doing its job well, you die from liver failure. Other organs are more resistant. Like you push them with reprogramming, but they still keep doing their job, and you don't you don't die, and the organs that can tolerate more reprogramming and potentially can get more rejuvenation from being pushed. So you push the brain farther, maybe than say kidneys. Kidneys you might want to push a little less far, other organs, and so you can have a very tailored combination with tailored durations of partial reprogramming eventually in this combinatorial combination therapy that we envision that can give the systemic rejuvenation if we target multiple organs.
What about I mean, if you have to be very careful with the liver, for example, I mean, the liver ages as well, so don't you have to reprogram it a little bit to actually get some rejuvenation effect?
It depends. Actually, the liver is the most or the only regenerative organ that we have. But I guess in case if you get some sort of actual disease or some insult to the liver, you potentially could do something to with partial reprogramming or other approaches to try to restore its function or even rejuvenate it. And some companies actually I know are targeting the liver for rejuvenation. I mean, Retro has been pretty open that the liver is one of the target organs that they're targeting with their partial reprogramming program. But I'm sure they're doing it in a very safe manner, not most likely not using 4 Yamanaka factors, and also maybe even using different reprogramming factors, transcription factors that are maybe liver specific. They only ever reprogram cells to like liver progenitor cells. I don't know. I don't want to speculate, but I'm sure they're being careful to make sure that they don't get liver failure. And another approach that you could do is, and with gene therapy, it's actually a potentially nice, no, not side effect, but nice aspect of gene therapy is that it doesn't target all cells. Like the efficiency of gene therapy in terms of delivery is actually like nowhere near 100%, and actually it's a good result if you get like 50% of your cells in a given tissue targeted. So potentially for the liver, if you use a gene therapy, if it doesn't target a high percentage of the cell, even if the cells that you are reprogramming, if they're kind of pushed a little bit too far to stop doing their work, the cells that you haven't transduced with your gene therapy, they actually keep doing the work, and you don't have liver failure. So that actually could be a good aspect of a gene therapy inefficient delivery. But of course, and if you actually do rejuvenate the cells that you have targeted, say 20% of your liver cells, potentially that could have some paracrine effects to the neighboring cells that could improve in general your organ functioning. And potentially you can envision having some like multiple bouts of targeting a given organ with different vectors targeting different areas of the organ, so that you can induce different parts of the organ or rejuvenate different parts of the organ at different times, so as not to have this overwhelming effect in case something goes wrong and your liver still keeps functioning while you are inducing partial reprogramming in some other area of your liver.
Now that you mentioned paracrine effect, I'm thinking of more broadly systemic effects, and there's this idea, or the skeptics say that partial reprogramming could just be one of the solutions to solving aging, because this is something that happens in the cells, and you have the extracellular matrix, you have systemic effects that cells don't necessarily account for, and so on. What is your response to that, and how is your thinking around that?
I actually just had this debate with Jean Hebert about partial reprogramming versus replacement. It's on YouTube. If you're interested, just Google debate.
You don't want to go through it again.
Well, yeah.
Maybe you could give the short version.
Oh, no. I mean, yeah, it's not that I don't want to go to it. I was just kind of shilling this video. And this debate has been going on, at least for me, with the proponents of the matrix hypothesis of aging, or the ECM, or like long-lived molecule degradation theory of aging. This has been going on for many years. probably close to a decade, because one of the first originators of this hypothesis is Alexander Fedintsev, and we've been debating this for many, many years with him. He's kind of the original ECM proponent. He's the guy who actually introduced it as a 10th hallmark of aging, the matrix, or I guess more broadly, the non-enzymatic degradation of long-lived molecules. That's like the general term for, I guess, stochastic degradation of, well, I guess the matrix is probably the biggest part of this category, but also, I guess, maybe even within the cells, Alexander mentions like nuclear pore complexes that can have some sort of stochastic changes that are irreversible. Basically irreversible because there's no enzymes within the body that can reverse this degradation. But I mean, broadly, I think the ECM, I don't think it plays a key role in aging. I mean, obviously, we do see this processes that make it stiffen and this cross-linking that we observe. But of course, when the body needs to remodel the ECM, or cancer is very good at remodeling the ECM. So obviously, the potential, the body still has the capability to remodel it. And also, within partial reprogramming results, when we look at tissue histology, we see definitely, even at the level of just, like the tissue histology, not just the individual cells, but the tissue, including the matrix, the tissue looks better. And there's a recent result in partial reprogramming of the brain, surprisingly, that actually looked at the ECM after partial reprogramming and reported that there are some aspects of the ECM post partial reprogramming are actually looking better. So there could be paracrine effects or effects on the surrounding matrix of these cells that we reprogram that could potentially be beneficial just through partial reprogramming. Whether or not, overall, as this ultimate therapy that will allow us to completely defeat aging, if partial reprogramming could be enough or you need something else, it's still an open question. And I mean, I'm not dogmatic. If eventually we discover that you actually need to do something about the ECM, or also I'm completely supportive of the replacement paradigm in itself. I think like if we're able to demonstrate that we can successfully transplant our head to a new body and we'll get full rejuvenation, the new body will remain young even despite our old brain, and then we can actually gradually replace the old brain with new tissue, which is of course Jean Abert's kind of main approach. Great, until then, I'd rather keep my head on this body and not risk transplantation until it's been fully proven. I'd rather have a gene therapy to try to rejuvenate at least the key organs of my body, which I think definitely at this point is a much safer paradigm and much more proven paradigm, because by now there's been over a dozen different studies showing potential beneficial effects of partial reprogramming in the context of diseases, in the context of lifespan increase. Whereas for replacement, I don't think we have had any successful study that showed that could be long-term, the new heads on old bodies, or vice versa. Actually, old heads on new bodies can have a sustained effect and have an increased lifespan, or can even remain without an immune rejection for a long enough period. Not to say that, again, I'm all for research in that direction, but I think we have to be cognizant of all of the problems that you might face with this transplantation of your head to a new body.
Talking about heads and brains, how is it going for YouthBio now? And you mentioned that you're focused on Alzheimer's disease, and I think that it's very personal for you, right?
My personal kind of Alzheimer's story is that my grandmother, she developed Alzheimer's and passed away from Alzheimer's many years ago. And I did a 23andMe DNA test. I discovered that I have 2 copies of the ApoE4 allele, which predisposes me, or at least greatly increases my risk of Alzheimer's, like 15 to 20 times, depending on which study you look at. And so, yeah, Alzheimer's been definitely a personal quest for me, or working on drug development against Alzheimer's has been a personal quest. In terms of
how long have you known about this, and what was your reaction? How did you handle that?
Well, I wasn't I wasn't thrilled to put it mildly when I discovered this. Also, of course, one other sad kind of downstream implication of this is that both my parents have at least one copy of ApoE4. If I have two, then both of them have at least one. And having my grandmother actually succumb to Alzheimer's, it's not looking good in terms of risk for my parents as well. And so I'm very cognizant of that, and just kind of on the lookout of how their cognition is progressing as they age. And definitely I'd love to have therapies against Alzheimer's be ready in time for my parents to use as well, while thankfully they're still okay. But in terms of how long, it's been a while. Like my grandmother, she passed away in 2011. So, and we knew that she had dementia she struggled with it for like 8 years, I think. And eventually, like, Alzheimer's was the ultimate diagnosis a few years after we started noticing the symptoms. So, and by the end of her life, she was like a completely different, not even a different person. I mean, just as I'm sure many people who have loved ones with Alzheimer's understand, just a very, very, she's no longer there, right? It's just it's a shell of a person that they used to be, and it's a very sad, very sad. To see someone you loved and knew, and I was quite close to my grandmother, because she essentially she raised me. When I was a kid, I spent many summers with her in Ukraine, because my parents would send me and my sister for the summer back in Soviet times. But it definitely was very, very sad. And of course, then from a kind of a selfish standpoint, once I discovered that I'm also kind of facing the same prospects, I wanted to see if I can in any way do something about it. And maybe it's, I think we all have these kind of, I guess maybe a little delusional that, I'm gonna solve Alzheimer's, but if you don't try, I guess you never succeed. But actually, I think like YouthBio from the very beginning was formed, as I mentioned, with this idea of tissue specificity, and the brain being the primary organ that we wanted to target with tissue specific therapies. And within the brain, Alzheimer's is a great candidate, not just for kind of my selfish passion against Alzheimer's reason, but also from a standpoint of like scientific reasons. There's recently, recently, but at that time that we were founding YouthBio, but now it's been a few years, there's some findings of a very strong epigenetic component to Alzheimer's, to the neurons that are affected by Alzheimer's. And so this is epigenetic driver of Alzheimer's. And as I said, partial reprogramming is a great candidate for epigenetic changes, because partial reprogramming essentially epigenetically rewinds the cell. We've discussed this in the beginning. And so we think just from that standpoint, partial reprogramming has a good chance to be a therapeutically beneficial approach against Alzheimer's. And also from the standpoint from a regulatory standpoint, unmet clinical need standpoint, Alzheimer's we think is a good indication, because huge unmet clinical need, huge number of patients, and the risk tolerance of the regulator, like the FDA, for example, is high enough for them to be able to allow testing of these new therapies, new gene therapies like ours. Because there's already precedent, the FDA has allowed other gene therapies, one of which is actually ApoE2 gene therapies, which I've been observing with quite high interest. And and also before our studies in Alzheimer's using partial reprogramming, there were some indications in the literature of potential beneficial effects in Alzheimer's mouse models using other epigenetic modulation, epigenetic regulation within neurons that were able to. One study showed the prevention of Alzheimer's symptoms, and another study showed even epigenetic modulation that reversed Alzheimer's symptoms, which was very encouraging. And now we have then started our partial reprogramming studies in mice, in the mouse model of Alzheimer's. We also now observe beneficial effects of partial reprogramming in preventing Alzheimer's symptoms. And also since then, like we had our results last year, we put a patent around it, and then Altos Labs came out with their results in a similar modality in a different Alzheimer's mouse model, but also neuron-specific partial reprogramming in the hippocampus with, I think, a 3-day on, 4-day off induction of periodic induction, weekly induction of partial reprogramming. They also observed the prevention of Alzheimer's symptoms, both in terms of cognitive tests and in terms of biomarkers like beta amyloid, which is very encouraging. And now David Sinclair also mentioned that research out of his lab, that at least started in his lab, has observed benefits in an Alzheimer's mouse model. He claims in reversal of Alzheimer's symptoms in a mouse model. So by now, I think there is several compelling kind of data points in terms of partial reprogramming and Alzheimer's that I feel very comfortable and confident and optimistic that at least we can start testing in humans this approach, partial reprogramming in humans, which is our next milestone in YouthBio. We really want to test this in humans, get to a clinical trial where we can test whether partial reprogramming can prevent the onset of Alzheimer's symptoms, or at least slow it down. And if we can reverse it, that would be the holy grail. If we can reverse Alzheimer's with partial reprogramming, that would be amazing.
What obstacles do you have to overcome to get to actually trial this in humans?
We need just a standard package of IND-enabling studies, safety studies, essentially. We need to demonstrate that the construct that we plan to give to humans, to inject into humans, is safe enough in animals that we can justify putting humans at risk of trying this therapy. And for that, we need funding. We haven't yet started raising the necessary amount that is required to get to that stage, to get this IND stage of essentially filing the application to start a clinical trial in Alzheimer's. So for the next 3 years, we plan to be doing these animal studies, this GMP manufacturing necessary to produce this gene therapy that can that will be used in humans. And for that, we plan to start a fundraise in the next few weeks to get this funding that will allow us to get there. So that's the main challenge. You have to do the animal studies, and for that you need the funding. So I guess funding is the biggest challenge, as it is for so many longevity companies and in general biotech companies, especially in the past few years. Funding has been a challenge.
It's interesting because when you see the amount of funding that Altos got, you would imagine that you would have. I mean, is it easier or harder to get funding with all the attention that Altos got and the reprogramming field as a whole?
I think it's actually harder, at least for us, because it's not just Altos. There's a few other companies with large investments, famously publicly known large investment sums, and investors. We've gotten quite a lot of feedback from investors who are like, I mean, we really like what you guys are doing, but we're just, afraid. There's Altos and there's Retro and other companies with much more money than you guys. So, we're not very comfortable investing in another company going up against the $3 billion purse of Altos. But then, to them, I usually reply that the field is large enough to support, dozens of different companies. If not just a single dozen, there's barely a dozen of partial reprogramming companies, because there's so many different diseases. And in terms of the brain, I mean, there's not a lot of companies targeting the brain therapeutically. I know Altos did its academic study in mice using an Alzheimer's mouse model, but they didn't use a therapeutic vector. They still used a transgenic mouse model in which they delivered an activator. And to my knowledge, we're the only company actually pursuing the brain as an indication therapeutically with our gene therapies. So,
speaking of the problems here, or obstacles, what are the main scientific obstacles that you see to use partial reprogramming as a way to slow aging or reverse aging?
well, I mean, I wouldn't call them obstacles. I think there's areas for improvement that I think for reprogram, because I think partial reprogramming is already ready to be translated to be used as therapies. Even in the current modality, you have to do it on a repeated basis. And obviously, safety is the main concern. But now we, first and foremost, with partial reprogramming, we know it's safe enough if you limit the expression to a short duration. Even in a transgenic mouse model where every cell has a copy of this cassette and you just express it for 2 days, the mice are fine. This has been tested not multiple times, not just in progeric animals, but in wild-type animals. First, Ocampo tested this in 2016, where they used 35 consecutive weeks inducing Yamanaka factors in normal wild-type mice for 2 days, 2 days on, 5 days off. That was safe. Then Belmonte group came out with a paper where they did this for 10 months consecutive weekly induction. Safe, no problems, no teratomas. Actually, they saw benefits in various tissues, like therapeutic benefits of partial reprogramming. So we know it's safe enough if you do it in a short duration. And of course, this was transgenic animals. As I said, every cell had a copy. We're using a gene therapy approach where we're only targeting a percentage of cells. And so even if something goes wrong and we keep overexpressing Yamanaka factors for too long, we're only going to affect a portion of the cells, not all of the cells in a given organ. And I think it's a safe enough modality that it's ready to be tried in humans. In terms of, as I said, room for improvement, right now you have to do it in a periodic manner. You have to keep activating Yamanaka factors like on a weekly basis in mice. I don't know what we have to do in humans, maybe bi-weekly, maybe monthly. And this is actually something that I think would be great to somehow either move away from. Like if we could induce partial reprogramming once and it rejuvenates the organism and it stays there for like months and months, that'd be ideal. Right now, what we see is once we stop partial reprogramming, we have this rebound, I call it rejuvenation rebound, post-partial reprogramming, that happens, I think, relatively quickly. And this is actually a bit of a under-researched area of what exactly is the dynamics of this rebound post-partial reprogramming. And this is
that you call it a rebound, but it sounds like it's accelerated aging, or is it not?
I don't know if I call. I mean, it's not really. I don't know. You could. I guess you could call it accelerate, or it's basically return to baseline. Whether it's accelerate or not, just how does it happen? I think what happens after you stop reprogramming is that the epigenetic pattern quickly returns to the original pattern. And some of the rejuvenation that happens is actually happening on this return to baseline, return to the original state. And we're still not sure what is actually responsible for this epigenetic memory. Some people call this epigenetic memory, and how it happens. And some groups actually reported this rejuvenation. The bulk of rejuvenation of partial reprogramming actually occurs during this period of like relapse. And so it would be really interesting to actually study the mechanisms responsible for this return to baseline and whether we can somehow decouple the mechanism of return to baseline of like the aging component of this baseline versus just the other gene expression, like the clusters of genes responsible for the function of the cell, like the actual cell identity determining cell clusters that also, like with reprogramming, they start getting suppressed, but after you stop reprogramming, they return to baseline. And like the fibroblast gene clusters, that was one of the area studied. They during reprogramming, they gradually get silenced, And if you kind of push it beyond the point of no return, they get silenced for good, and you just go on to the pluripotency pathway. But then if you stop reprogramming, then they gradually return back to the baseline, and you get back to this fully fibroblast state. But also there's some aging components, and you could look at this methylation clock, and it'd be great to actually decouple these processes and kind of keep the identity gene clusters at baseline, but only suppress the aging cluster genes, or only change the pattern of the gene expression that is responsible for the aging effects. So that's, I guess that's a future direction for the entire partial reprogramming field, and the area for improvement, that if we're able to successfully figure it out and find ways to modulate it, then we'll be able to move away from having to do reprogramming on a repeated basis, or at least greatly decrease the frequency with which you need to induce reprogramming to kind of keep your biological age at a lower level, or not, or prevent its increase over time. So in terms of obstacles, well, I mean, in general, also, I guess for gene therapies, delivery is a bit of a challenge. Delivery to also definitely a tissue-specific delivery, like only targeting the organs that you want without targeting the cell types that you don't want. And of course, we circumvent it with using our cell type-specific promoters, that even if our vector ends up in the cell types that we don't want to have an expression in, the cell type specific promoter will ensure that there won't be expression in those cell types, that we only get expression in the cell types that we target. And also, we can also have, as a next step in our development, specific targeting vectors that have preferential tropism to the cell types that we want. This is a kind of additional safety layer for the future, again, like the room for growth. But even with the current technology, we can already, I think, have therapeutically safe and efficient gene therapies that we can start testing in the context of patients that already have diseases, and so the risk tolerance is high enough to try these novel therapies. And during the course of this research, we'll also establish just how safe it is in humans, just how safe it is for long-term application in humans, and we'll iterate from there and try to improve it even further.
I was watching one of your talks, and you were talking about certain ants and how they're able to decelerate their rate of aging. Observing these ants and similar organisms, is that something you learn from?
Oh, this is just one example out of probably maybe a dozen different examples in nature that I see for the potential of epigenetic modulation of lifespan. And these ants, the example you speak of is Indian jumping ant, in which you have different social roles. Of course, there's the queen, the breeder, that has the longest lifespan, several years. And then you have the workers that have the shortest lifespan of the social roles. Of course, they're twins. They're all identical DNA. It just depends on the social role which they end up with. Their lifespan gets kind of assigned correspondingly. But they also have this ability, this particular species of ant, if the queen dies, some of the worker ants are able to switch their social role into a breeder, and they have the gamma gates, this special like in-between social role, which by default they're workers with a default short worker lifespan. But if the queen dies and they need to actually become breeders, then they epigenetically kind of switch their role and they get a much longer lifespan. And to me, this is just showing, even in the context of a single organism, there are abilities for the genes to greatly modulate lifespan epigenetically. And this is proof of epigenetic control of lifespan that we observe in nature. And then it's, to me, very reasonable to assume that in mammals, This is also what's happening. It's epigenetically. Life history, including the lifespan, is implemented epigenetically. But beyond ants, another example I like in mammals is we have this montane voles, these small rodents that live in Montana, that have also they could have a longer lifespan depending on the season in which they are born, which again to me showing that there's epigenetic modulation of lifespan even in mammals, even in the context of a single animal. If they're born in the spring, that they quickly mature within the same season. In the summer, they breed and they die within the same year. But if they're born in the fall, they slow down their development. They actually pause their development until after the winter, and they get sexually mature after the winter, and they have a correspondingly longer lifespan. They basically die next year. And so this shows to me that there is, like, if you can hack epigenetics, if you can like freeze the epigenetic state of an organism in most of the cells or all of the cells, then you can potentially greatly increase lifespan that way.
I mean, it also happens for the whole organism. They slow the aging rate for the whole organism epigenetically somehow. So I suppose one could get a lot out of studying the exact mechanisms in these animals.
Yeah, definitely. I think this is also an understudied area of research in our field, and just how the potential central regulator that synchronizes all of these processes is implemented. Because obviously we have a central regulator that controls our growth rate, that controls the stages of our development, controls obviously our circadian rhythms, controls our periods for females, right? Menstrual cycle, estrous cycle, if we're talking about other mammals. So there is a very obvious regulation and synchronization for various life stages, but for some reason, for aging, it's actually, I think, within the field, not accepted that there is any sort of synchronization for aging. They think, some people think it's completely random. There's no, but there obviously is for other developmental stages, but then studying at least those developmental stages, I think, could be very useful in the context of then trying to see, does this synchronization process continue into the aging stage of our life history?
In your clinical trials, are you looking to have a cohort of patients with where you actually try to prevent Alzheimer's, or are you going to have a cohort where you're actually trying to reverse the symptoms? What do you think about that?
Kind of the open question right now that we're debating, and we still haven't finalized the protocol, the clinical trial protocol that we'd love to have. But I think, yeah, in the beginning, we'll start with patients. Most likely, we'll start with patients that have already some sort of like maybe mild cognitive impairment or early stages of Alzheimer's, and we try to see if we're able to prevent or slow down, or if we can reverse it at that stage, great decline into Alzheimer's. And another alternative is if we start with patients with advanced Alzheimer's, seeing if we can reverse it. But of course, I guess an argument could be made that by that stage, it might be too late. So, and obviously, if we can prevention is probably kind of the first step. If you can prevent it, it's already a good result. And if you can prevent it, then you can kind of look at the next challenge. Because if you start from people with advanced Alzheimer's hoping to reverse it, that might be a bit of a too risky of a bet to take it in the beginning. So you might like do the progression from people from prevention to reversal rather than the other way around.
There is always this debate about health span versus lifespan. Should we try to focus on prolonging the healthy period of life and then die, or should we actually try to solve aging and be able to live a lot longer? And I know you're probably more of a lifespanner than a health spanner.
I don't. Anybody who says we should then die, like immediately that completely triggers me. I'm like, why? I don't like anybody saying, "Oh, you just die." It's like then you should just die, or people with they should just die, or they should just accept the death. I don't agree with. I think any death is a tragedy at any age, at any stage. It doesn't matter, or any kind of health level. And I think our goal, as is the goal of medicine, I think, is to prevent death and to have people not die, have people, if they're sick, get healthy. If they're healthy, remain healthy for as long as possible, as long as they want. So to me, this kind of juxtaposition of health span versus lifespan seems just a PR move to people who are afraid to claim that they're working on extending lifespan, because this has a sort of a negative PR attached to it that I don't even know how it got to this point, but somehow the general public thinks it's a bad thing that some people are going to live longer, and if the bad PR stands for like billionaires are going to live longer. Oh my God, it's so unfair. They're already billionaires. Let them die at least as regular people. And so the kind of the answer to that was that for some people to say, oh, we're not trying to extend lifespan. We're only trying to get people to die healthy. So we're extending health span and then let them drop dead at age 85, but healthy, which in itself is a complete contradiction, because if people are healthy, they don't die. Healthy people don't die. People die at 85 because they're very sick. If you're 85 and your health is that of a 20-year-old, you're not going to die. You're going to live and be as vibrant and healthy as a 20-year-old. So that whole idea that you can somehow increase health span and then people are just going to drop dead for some unknown reason at age 85 being completely healthy is just ridiculous. And so, I mean, but that said, obviously I want to, like, as a small first step, if we can increase health span with existing known interventions like rapamycin, metformin, and get people to use them, that will make them like healthier at current, age 60 than people who are not using them are currently, great. We should definitely do that. We should always aim to get people as healthy as possible. And if the intervention is not really increasing maximal lifespan or increasing it a little bit, but it's increasing health span more significantly, I'm all for it. It's a great intervention. But as a goal, I think we as a field are definitely striving to get people to be as healthy as long as possible. And so ultimately, yeah, we're all working against aging. We're all working against death and the causes of this death. And having somehow be embarrassed that we're trying to eradicate aging and death is, I think, again, is ridiculous. Somehow people feel ashamed that, we're trying to make people not die. And I think also in society in general, I think there's some aversion to the word death, when, at least in the context of aging, when they hear that you're against death or something about death, then they get all of a sudden this emotional reaction and think that you're doing something wrong. But I don't want to hear about death. Don't talk to me about death. Like, you're trying to cure diseases, great. You're trying to stop death, you're somehow a weird person. So sorry to go on this rant, but as I said, I get triggered by this health span is good. And like,
why do you think? I mean, I really appreciate your honesty, and I mean, both me and Patrick, of course, agrees with you on everything you just said. But why do you think that so much of this aging biology field are afraid to speak out on this? What's the cost associated with actually being honest here?
well, I think it's a bit of a group speak, and just I think most people are afraid to be kind of the odd one out, and there's already this kind of academic, very mild kind of like you don't want to make bold claims, or at least in terms of goals, you don't want to anger the society at large. I don't know. And once new people get into the field, they just take on the kind of the existing ethos of the field. And if people in the field already have been saying for decades that we're really about health span, and we're not really about living forever, and then new researchers just kind of keep going with the same theme, not to anger their PIs or grant giving agencies or whatever. They just keep following the prevailing field mantra, right? So maybe that's why. And
so how much of the science of reprogramming is explicitly anti-aging, and how much is not? Is just focusing on various cures?
Well, I think ultimately like what we're doing is about anti-aging, or about combating aging. And from the beginning, the paradigm of partial reprogramming was a paradigm of combating the changes that epigenetic changes that drive aging. And of course, the first study was geared towards extending lifespan, so preventing aging or slowing aging down to extend lifespan. And of course, these studies are long and expensive, and also for translational purposes, to get something on the market, you have to show efficacy in a disease. Like the FDA, if you go to the FDA and say, oh, we're going to have a gene therapy that targets all your cells and makes you live longer, I don't know, in 30 years, if we run a clinical trial now on like 50-year-old people, they're not, first of all, they're probably not going to allow this trial. And second, you're not going to be able to do this clinical trial just financially, or so you need an indication in which you can show a benefit in a reasonably short time span, like a year or 2 years, so that this is what drug development model is geared towards. Because if your clinical trial lasts 10 years, obviously you're financially just never going to make sense. And so this is how you translate even interventions against aging. You find a disease in which it's effective, you get it on the market, and then you can start expanding that label, the drug label, to include other diseases. You run other clinical trials. In terms of partial reprogramming, what we envision is having therapies against multiple organs, demonstrating benefit in the brain, then target some other organs, demonstrate benefit then in those organs. And once multiple therapies for multiple organs are on the market, you can start combining them and potentially running a trial that tries to prevent like multiple morbidities. So something similar to the TAME trial, which again actually is the closest thing to slowing down aging as I can imagine right now, in terms of like the FDA type of trial. But overall, like partial reprogramming to me is going after aging, is going after the key driver of aging. But translationally, to get it on the market, to get these therapies on the market, you need to do this for some existing diseases first. So.
Like we have quite a few exciting years ahead of us, with several clinical trials happening before 2030.
Oh, absolutely. Yeah. Like next year, if YouthBio succeeds in starting their clinical trial, they should have the NION, the eye stroke clinical trial. Altos Labs reported that they're already trying something in the clinic with kidney transplants, that they're trying to rejuvenate transplanted kidneys by partial reprogramming. So they're already looking at some clinical applications. A few other partial reprogramming companies that I'm aware of are also close to, like within a year or two of the clinic. TurnBio with their mRNAs, I think they're targeting skin or T cells, like ex vivo rejuvenation of T cells. And with the brain, we're targeting Alzheimer's. Not sure any other companies like clinically targeting the brain, but who knows? Maybe we'll have some others. And I think the next 3, 4, 5 years are going to be exciting in terms of partial reprogramming. By 2030, yeah, I'm very optimistic that there'll be already some approved partial reprogramming therapies, and we'll have future next generation therapies that are even safer, they're more targeted, and potentially with different factors, like not Yamanaka factors, but actually like cell type specific combinations of factors that could produce even better rejuvenation or more durable rejuvenation for that specific cell type. So I'm very optimistic.
So Yuri, thank you so much for taking the time to talk to us today. As always with our guests, we are so humble and we're learning so much. So it was wonderful, and as I said in the beginning, your particular field is so exciting and promising for the future. So where can people get in touch with you if they want to maybe come help you out with funding, for example, or anything else?
Yeah, that'd be great. Anywhere, I mean, I'm on LinkedIn, Twitter, or YouthBioTX.com is the website for the company, YouthBio Therapeutics. So yeah, just you can contact through there or through my DMs are open through any of the social media platforms. So
Perfect.
anything
Hope
works.
we'll talk to you again sometime.
Yeah, and thank you so much for the invitation. And it's a very topic that's very dear to my heart. So I could talk for hours. And thank you for giving me the chance to do so.